Next, we need to deal with the anatomy and histology of the accessory glandular structures that serve as para-alimentary structures associated with the gut tube. And our conversation begins on this slide with the differences in the secretory products and nature of the three salivary glands. The parotid gland is unique among the three in that it is virtually entirely a serous-producing salivary gland. And each salivary gland that's parotid basically empties by way of stensons or the parotid duct that opens into the oral cavity above the second upper molar tooth. Parotid gland secretions basically account for about 25% of the volume of saliva. Both submandibular and the sublingual salivary glands are mixed glands. The submandibular has a predominance of serous cells, whereas the sublingual gland seems to have more mucus cells. The submandibular glands are the major salivary glands in that they contribute to about 70% of the volume of saliva. The sublingual glands are the least significant of the salivary glands in that their mixed secretions constitute only about 5% of saliva volume. Histologically, the section that's shown on the next page shows a mixed gland that has to be either one that's either submandibular or sublingual. And basically, we've identified it as being submandibular, although you would have to be cautious in your ability to evaluate that. But aside from that feature, you can certainly see that it has to be one or the other because it contains a mix of lightly staining mucus acini that are closely related to the much more darkly staining serous acini. And you can see where there is a mucus acinus, typically you see what are known as serous demilunes, where, because of histologic processing, the darker staining serous cells are pushed to the exterior of the grape-like acinus structure. The arrowheads point to small blood vessels, but the other point to be made is there's a slightly eosinophilic duct, an intercalated duct or striated duct, that's situated near the upper left corner of the slide. Next, let's look at the anatomy of the exocrine and with it the endocrine pancreas. The exocrine pancreas is obviously involved in the secretion of many different kinds of enzymes we'll talk about in a moment. But you can see that the pancreatic histology is easy to recognize. Aside from the very deep basophilic staining cells that make up its exocrine portion, certainly interspersed among the lobules of the pancreatic exocrine acini are the islets of Langerhans, which are shown at the arrows. These are pale staining regions that are dominated by the beta cells that produce insulin. And the insulin cells we know are not only about 70% of the islet of Langerhans cell types, they are mainly found in the central region of each islet. More peripherally located are the glucagon-producing cells and the somatostatin-producing cells. But with specialized histological staining techniques, you cannot distinguish an alpha, beta, and a delta cell from each other, except on the basis of presumed location. Next, what are the major pancreatic exocrine secretions? One of the major of them is bicarbonate, and actually bicarbonate is produced by the pancreatic duct cells, not by the pancreatic zymogen-containing secretory cells. So the bicarbonate ions, basically their job is to neutralize the hydrochloric acid medium of the chyme that is entering the duodenum from the stomach. This bicarbonate also serves to deactivate pepsin. So when basically the acidic medium enters the duodenum, the S cells, which are enteroendocrine cells in the lining epithelium of the duodenum, release secretin, and that, as we say, acts on the pancreatic duct cells to increase their bicarbonate production and output. And we know that bicarbonate is produced by the action of carbonic anhydrase on carbon dioxide and water in the pancreatic duct cells. Next, what are the overall pancreatic secretory products in addition to bicarbonate? Well, the pancreatic enzymes that are stored in the form of inactive zymogen granules constitute 15 different enzymes at least, and they are responsible overall for the digestion of proteins, carbohydrates, lipids, and nucleic acids. And when small peptides, amino acids, and fatty acids actually enter the lumen of the duodenum, cholecystokinin is released by enteroendocrine cells that are, again, in the lining epithelium of the duodenum, and cholecystokinin basically stimulates pancreatic enzyme secretion. And this secretory activity is enhanced by vagal stimulation by the release of acetylcholine as a neurotransmitter that potentiates the action of secretin and stimulates enzyme secretion as well. Certainly, those pancreatic acinar cells have to be protected against self-digestion, and as a reason for that, all these proteolytic enzymes are stored as inactive precursors in what are known as zymogen granules. Next gland we need to worry about the structure of is a very important one, and that's the liver. In a way like the pancreas, has a dual role. It certainly has an exocrine product in the form of bile, like the pancreatic enzymes, and it certainly is involved in a lot of endocrine-type secretion, release of mainly proteins like albumin into the bloodstream. So fundamentally, the basic structure of the liver can be organized in a number of anatomic ways. One of the classic ways to illustrate liver organization is to talk about the organization of a liver lobule. And a liver lobule basically is in the form of a hexagon where the central element of a liver lobule is the central vein. And this liver lobule organization organizes a region of the liver, in this case the lobule, into a blood and bile flow functional diagram, in the sense that the central vein in the middle of the liver lobule basically is the recipient point of all of the blood that is entering at each of the 6 points of the liver hexagon. So at each of the 6 points are what we refer to as being a portal triad. And the portal triad, and the anatomy of it is illustrated on this picture. Each portal triad at each point of the hexagon contains a branch of the portal vein, and this portal venous blood, it basically is coming from the intestinal tract, and basically it constitutes or forms about 75% of the blood entering the liver lobule. The remaining 25% of portal lobular blood is entering by way of a branch of the hepatic artery. The third element of the triad is a tributary of the hepatic duct or bile duct, as illustrated on this page. So the point is, blood is flowing from the perimeter of each point of the liver hexagon toward the central vein, but bile, on the other hand, that's the exocrine product of liver hepatocytes, is flowing in the opposite direction, away from the central vein toward the lumen of a tributary of the biliary apparatus at each point of the liver hexagon. The inset on the lower right shows the structure of a liver sinusoid. The sinusoids are specialized or modified blood vessels that are lined by a discontinuous fenestrated endothelium, and that endothelium basically is interspersed by Kupffer cells. Kupffer cells are the macrophages of the liver that basically are involved in take up, among other things, of blood cell remaining products, including iron. But in essence, they are liver phagocytes that will basically examine and phagocytose any luminal contents of the liver sinusoid. Note that on the outside of the liver sinusoid, between the endothelium and the Kupffer cells and the hepatocytes, is the space of Dease. And in the space of Dease, even though it's not illustrated here, are another specialized cell type in the liver known as an Ito cell. Those hepatic stellate or Ito cells, their major job is to store fat in the form of vitamin A. And in addition, you can see that outside the space of Dease are irregular plates of hepatocytes, and what you're seeing interposed between two of them, or three of them, are the bile canaliculi into which bile is released from a corresponding hepatocyte. And what we're showing in the direction of the black arrow is bile is obviously in the direction of the arrow flowing from left to right in this diagram, so that means that blood flow has to flow through the sinusoid from the right side of it over to the left, because in the liver lobule, blood flow is in the direction opposite to that of bile flow. Now, that's basically the classic hepatic lobule or liver lobule, basically, that has a hexagonal structure, a portal tract or triad at each corner of the hexagon, and blood is flowing toward the central vein in the middle of that hexagon. So that's the point that we made in summary, is that the blood is flowing toward the central vein from each of the 6 points of the triad, whereas bile is flowing when produced by the hepatocytes and released into the bile canaliculi from the portal vein in the direction of the portal triad at the perimeter of those hexagons. Now, in a histologic section, we can see some of the anatomy of basically the liver. And again, what you're looking at fundamentally at A is a central vein. And surrounding it, basically, although not in a typical form of a hexagon as shown here, is basically the anatomy of that liver lobule. So at each of the arrows is kind of a connective tissue trabecula that contains branches of the portal triad that are surrounding the lobule and delivering blood into the central portion of the lobule that will ultimately drain into A. So the structures at B represent locations of 2 of the portal triads, each of which, as we said, contains a branch of the portal vein, a branch of the hepatic artery, and a tributary of the biliary system. The other point that we want to make about liver organization is to talk about the structure of a hepatic acinus. And basically, this is a direct representation of blood flow that is progressing from the area at the perimeter of a liver lobule toward the central vein. And the hepatic sinus, instead of being hexagonal in its organization, is shaped like a grape, hence the term acinus. So this organization is exclusively related to direction of blood flow and organizes the regions of the hepatic acinus into 3 zones. Zone 1 will be basically the area that will receive the arterial and venous blood first. Zone 2 is an intermediate zone. Zone 3 is the region of the acinus that is closest to the central vein. So, and obviously, the hepatocytes, as you'll hear about in physiology, have vastly different functions depending on whether they are located in Zone 1, Zone 2, or Zone 3. And we just took the previous slide and we've illustrated on here numerically the 3 zonal regions that begin near the border of that liver lobule and extend in close proximity to the central vein. And certainly Zone 1 would be the area of the acinus that first receives the oxygenated blood. Zone 2 is the intermediate zone. Zone 3 hepatocytes, obviously, are going to be the last cells to receive oxygenated blood from the entering portal vein and hepatic artery branches. Finally, we made the comment earlier about the Ito cells, or the hepatic stellate cells. These cells are situated between the fenestrated lining epithelium of the sinusoids of the liver and between the microvilliated endocrine-producing surfaces of the hepatocytes. These Ito cells, as they're called, lie in that space of Dease, and basically they contain and store fat, and they're also involved in the storage of fat-soluble vitamins, particularly vitamin A. So these normally quiescent cells are there minding their own business by storing these elements, but when they are stimulated during liver injury, such as in a patient that is a chronic misuser of alcohol, the Ito cells may transform into a form of a fibroblast and produce and secrete type 1 collagen and other matrix component into the space of Dease. This contributes to a scarring and a blockade of various sinusoids in the liver, and obviously is a leading precipitating cause of the development of portal hypertension and the related portocaval anastomoses that are frequently associated with either esophageal or rectal bleeding. Last but not least, let's talk about the exocrine function of the liver, and this certainly is an emphasis on bile formation. So bile, basically salts, are secreted into the lumen of the duodenum, and they aid in fat emulsification and absorption, as well as in being involved in the excretion of exogenous metabolites like bilirubin and drug metabolites that cannot be excreted by the kidney. So bile consists of a mix of bile salts, which are mainly conjugated bile acids, conjugated bilirubin, and other endogenous drug metabolites, and they also include cholesterol, phospholipids, electrolytes, and water. Next slide illustrates the function of the gallbladder. Fundamentally, it does 2 things. It not only stores bile, but it concentrates it. And the mucosal lining of the gallbladder looks somewhat similar to the lining of the small intestine in that it's lined by a simple columnar epithelium, but that is the only cell type that constitutes the mucosal lining of the gallbladder. And that simple epithelium, the columnar epithelium, therefore has both absorptive and mucin secretory functions. It's related certainly to a folded underlying lamina propria, but unlike the rest of the intestinal tract, the gallbladder lacks a muscularis mucosa and a submucosa, and so that directly underlying the lamina propria will be layers of smooth muscle that obviously facilitate bile release. The entire surface of the gallbladder is covered by a serosa because it is a peritoneal structure. And fundamentally, the largest component of the gallbladder is the fundus. It has a narrowing neck. And the unique feature about the cystic duct that joins with the hepatic duct to form the bile duct, basically in the hepatoduodenal ligament, the cystic duct is a two-way street because bile that's been released from the hepatic ducts can pass through the cystic duct into the gallbladder for storage and concentration. But then upon cholecystokinin release, bile then must pass down from the gallbladder through the cystic duct into the bile duct so that it can enter the lumen of the duodenum. Our last histologic section of the gastrointestinal chapter shows a section that illustrates the mucosal lining of the gallbladder. Note that the lining, including the lamina propria and its epithelium, are thrown into very thin folds that are certainly much thinner and more likely to be branched than villi that characterize the small intestine. But again, the epithelial lining is exclusively that of a single cell type, a simple columnar epithelium with no intervening goblet cells. Underneath the epithelium is an underlying lamina propria, but as we said, there's no muscularis mucosa, nor is there a submucosa. So directly underlying the lamina propria will be smooth muscle and an associated serosal layer of mesothelium.